Hello everyone, my name is Jacklyn Staffeld. I'm a third-year student at Eastern Michigan University, and I'm so thrilled to be here to share with you my presentation probing the role of the ventral hippocampus, the nucleus accumbens pathway, in individual learning. In the United States alone, there are 21 million people suffering from at least one addiction, leading to 93,000 overdose deaths in 2020 alone. Due to the growing societal impacts of addiction, addiction research has become critical in the scientific community. Even more concerning, however, is how some individuals can actively use addictive drugs and stop with relative ease, while others may try a small dose before becoming lifelong dependent. Research has begun to consider that some individuals may respond emotionally to environmental cues, creating emotionally salient events that may motivate their behavior. Through the use of animal models, it is possible to venture into the studies of these considerations and strive to find answers. The participants for the study originally included 32 Sprague Dawley rats, 16 female and 16 male. These rats were approximately 7 to 8 weeks old, purchased from Charles River Breeding Laboratories, and were put on a 12-12 dark light cycle in which the lights turn on at 7 p.m. Following surgery, the rats were 16 male and 15 female, one of the rats having to be excluded due to some post-operative complications. In order to use animals to model addiction, we use a Pavlovian conditioned approach, or PCA, procedure. In order to use animals to model addiction, rats are often classified using this procedure. PCA is a session in which rats are placed in a controlled environment in which an illuminated retractable lever is presented for a few seconds, followed by a food reward, the unconditioned stimulus. Although there is no need for the rats to engage the lever in any way in order to receive the food reward, they learn to associate the presence of the lever with the acquisition of reward. As PCA progresses, two learning behaviors are observed as the association between the two stimuli is made. Some rats, referred to as sign trackers, seem to treat the lever as an incentive in itself, sniffing, gnawing, and clawing at the lever when presented, as well as accepting the food reward. Goal trackers, on the other hand, seem to treat the lever as just a precursor for the food reward. These observations, we believe, make sign trackers more prone to addiction-like behaviors and relapse behaviors. We believe this because sign trackers have cue-induced seeking behaviors. In terms of drug use, this means that stimuli previously associated with the drug may cause drug-seeking behaviors, leading to more prone to relapse. Once we had determined this behavioral type, we wanted to see where in the brain this might be caused. Previous studies in our lab led us to the ventral hippocampus to nucleus accumbens projection. In our 2016 Fitzpatrick et al. Experiment, we considered how the hippocampus as well as its subregions may be involved in this process of sign tracking and goal tracking. The hippocampus is associated with a variety of memory types such as spatial navigation and encoding of contextual information. This may play an important role in addiction and the environmental aspects of usage. Also of interest was the increased level of dopamine within the nucleus accumbens for sign trackers. It has been previously found that the ventral hippocampus is critical for dopamine activity in the nucleus accumbens, and lesions of the ventral hippocampus region showed a decrease in dopamine levels in the nucleus accumbens. We also saw a decrease in sign tracking behaviors in response to the lesions of the ventral hippocampus. Similarly, lesions of the ventral hippocampus showed a decrease in the concentration of dopamine metabolite homovanillic acid. These findings suggest that the ventral hippocampus and nucleus accumbens may play an important role in the acquisition of sign tracking behaviors. Once we determined the area of the brain that we wanted to manipulate, we had to determine how we were going to manipulate it. Optogenetics have been an engineered method for researchers to interact with the molecular chemicals in the brain. Of the many classes of genetically engineered protein, designer receptors exclusively activated by designer drugs, or DREADs, are one of the most prominent. Designer receptors exclusively activated by designer drugs have been considered the most widely prominent because they are G-coupled mescarinic receptors that have been genetically modified to respond specifically to particular compounds that are foreign to the body. For example, in our study, we used an HM3DQ DREAD that is typically used to excite firing and activating within a particular pathway. This is strictly activated by clozapine N oxide, or CNO, a synthetic compound that is not naturally produced by the body. Once you inject CNO into the body, there is excitability for approximately 2 hours. To use our dreads, we perform stereotaxic surgery using an in vivo dual vector approach to bilaterally inject Cre accumbens into the nucleus accumbens and an excitatory Cre-dependent dread into the ventral hippocampus. This particular dread is Cre-dependent because it is only taken up into the genome if Cre is present. This allows us to selectively target the ventral hippocampus to nucleus accumbens projection. 26 rats were given the surgery, while the other 6 received a control surgery in which an M-cherry control was injected into the ventral hippocampus and the Cre was injected into the nucleus accumbens. The rats were then given a period of 5 weeks for the dread gene to be integrated into the genome and produce the desired receptors. We then had 6 days of PCA, followed by 1 day of crossover treatment and 2 days of conditioned reinforcement. Our findings show that there were no statistically significant differences amongst treatment groups for lever press numbers, latency, and probability in comparison to the goal tracking magazine entry numbers, latency, and probability. These findings concluded that the dread CNO excitation of the ventral hippocampus pathway had no statistically significant effect on acquisition for sign or goal tracking. However, although there were no statistically significant effects, we did see a trend in which the dread CNO group tends to sign track less frequently than the dread vehicle group. This suggests that there may be a difference in sign tracking behaviors requiring further investigation using an increased sample size to determine statistically significant effects. We also saw that there were no statistically significant effects in sign tracking lever presses or goal tracking magazine entries for either the dread CNO or the dread vehicle. These findings suggest that the expression of the sign and goal tracking behavior is not affected by different treatments following acquisition. Also of interest was the findings of the conditioned reinforcement test. Conditioned reinforcement uses an active and inactive nose poke port in which the lever is presented only in response to the active nose poke. Typical data suggests that the rats will have preference towards the active nose poke, sign trackers having particular interest in it due to the incentive salience that is associated with the lever. Data showed that the dread CNO rats showed statistically significant preference for the active nose poke. Interestingly, however, dread vehicle rats consisting of sign trackers and intermediates only did not show preference towards the active nose poke during the conditioned reinforcement. Despite the lack of statistically significant results in our experiment, we did see a trend in which dread CNO rats sign track less frequently than the dread vehicle rats during acquisition. These findings suggest that there may be a trend of increased activity in the ventral hippocampus to nucleus accumbens pathway, leading to an increase in sign tracking behaviors, a decrease, excuse me. This finding does not seem to parallel the 2016 experiment of Fitzpatrick et al., in which lesions of the ventral hippocampus showed a decrease in sign tracking behaviors. However, the data does follow previous studies in which lesions of the hippocampus showed an increase in sign tracking behaviors, as well as our previous inhibitory experiment in which statistically significant decreases were found in sign tracking behaviors during the expression test in which dread vehicle rats received CNO. These findings suggest that further investigation is necessary in order to determine the connection between the ventral hippocampus to nucleus accumbens pathway and sign tracking behaviors. Animal models show excellent promise for paralleling human addictive behaviors. These findings suggest that there may be a connection between increased activity in the ventral hippocampus pathway and a decrease in sign tracking behaviors. Should there be an association, whatever it may be, this may offer insight into the neurological causes of sign tracking behaviors, and through translation may offer an opportunity to research neurological aspects of addiction and allow for manipulation or treatment of learning and behavioral styles as research progresses. I'd like to thank you all for listening to my presentation and give a special thank you to my mentors, Dr. John Morrow, which is the head investigator for this project, Dr. Karen Solis, my co-mentor, helping me through this project, and Frankie Cizak, a graduate assistant that was leading me through this project. I also like to thank the McNair members and staff for all of their help through this project and all of the support that they've given me to pursue research. And I'd like to just thank the Undergraduate Symposium for this opportunity to present. Thank you.
Thank you, Jacklyn, and our mentors, Karen Solis and John Morrow, for guiding her very important topic. So does anybody have any question?
Yeah.
Why do you think for your treatment group there was the differences in the